Welcome to my presentation on challenges in using RoB2. Worked example from a systematic review of remote therapies for alcohol and drug misuse. This was presented recently at London Cochrane Colloquium 2023. Assessment of risk of bias is an integral part of systematic reviews evaluating effects of an intervention. The revised Cochrane tool RoB2 for assessing risk of bias is a substantially improved revision of the initial version, which aims to increase the reliability of assessments. RoB2 addresses a broader range of issues than the initial version, such as post-randomization deviations from intervention, assessing the risk of bias on a particular outcome, and incorporates recent developments in estimation of intervention effects and theoretical discussion of bias. The aim of this presentation is to share our experience of using RoB2 in the context of systematic review, how effective are interactive remote therapies for reducing drug or alcohol use. I would like to discuss the dimensions of the review. Participants were adults receiving treatment for drug or alcohol or mixed substance misuse from OECD countries. The intervention we were interested were delivered remotely or digitally, that is online or by telephone or through a mobile smartphone application. Types were remote recovery support, remote talking therapy, and self-guided therapy, which was delivered alongside, after, or instead of in-person treatment for drug or alcohol misuse. This was compared with any type of comparator, including in-person treatment, no intervention, or an alternative remotely delivered intervention. The outcomes we were interested in was any quantitative outcome measures of drug or alcohol consumption, such as days of use or relapse. We conducted 53 RoB2 assessments of 45 randomized controlled trials. 8 studies included 2 relevant outcomes which were each assessed separately for 2 substance use outcomes, such as relapse rate, that is failure to maintain abstinence, and days of consumption. Only 1 study was judged to have a low risk of bias. In the remaining 44 studies, risk of bias was judged to be high or as indicating some concerns. So the main focus of today's presentation is to discuss the challenges we faced. We faced 4 challenges. Pre-specifying the outcome to be assessed. Second, assessing and reporting multiple outcomes. Third, interpreting statistics and trial methodology. And fourth, difficulty in navigating the guidance. So let us see one by one. First challenge was pre-specifying the outcome. Why this was a challenge? The studies included in the review were heterogeneous, evaluating complex interventions with a range of outcomes. Currently, there is no consensus regarding core outcomes of substance use treatment interventions. No preferred outcome by the review commissioners. So what we did, this is the iterative process we have undertaken for selecting the outcomes. So we had 4 decision points. Decision point 1, we looked at outcomes relevant for stakeholders. Though they did not have a preference for a particular type of substance use outcome, but they were not interested in other outcomes such as mental health or criminal justice involvement. So we could eliminate looking at those type of outcomes. Decision point 2, commonly reported outcome. We engaged with all substance use outcomes across our data set of full included papers. Third decision point, we looked at the most robust measure of the outcome. So we prioritized toxicology or self-report verified by toxicology over self-report only when a study reported multiple eligible outcomes. Decision point 4, comparability of outcomes. We considered the type of measures such as dichotomous or continuous measure, type of statistics used, and also the outcome measurement time points. So, based on our experience, the following suggestions we would like to provide. Guidance should recognize that not all reviews include a homogeneous study set with a standardized outcome measure. We need to recognize and acknowledge that included studies in most reviews are heterogeneous, diverse, and complex in nature. As a pragmatic approach, reviewers could pre specify decision rules, for example, how they are selecting the most reported and relevant outcome. Reviewers could also familiarize with the literature prior to the systematic review, consult subject specialists and stakeholders. This will be helpful in selecting the relevant and commonly used or reported outcomes. The second challenge was assessing and reporting multiple outcomes. This was a challenge because studies with multiple eligible outcomes required time consuming and largely repetitive assessments. Although some aspects of RoB2 are specific to each outcome, others apply to the whole study. It would be preferable to have a system for assessing the RoB for studies with multiple outcomes in a way that avoids duplication of domains that remain the same for all outcomes. So what we did? We identified RoB2 items that are likely to change during multiple outcomes from a single study are assessed. So in context of review, domain 1 and domain 2, there was no change. In domain 3, with regard to missing outcome data, we had continuous measure and dichotomous outcomes. So continuous outcomes were treated differently. So we considered availability of data from 95% of the participants as sufficient for continuous outcomes. For dichotomous outcomes, when the observed number of events is much greater than the number of participants with missing outcome data, the bias we considered to be small. Domain 4, there was some changes. For self-report substance use outcomes, influence of interventions involved monitoring drug use, which could affect measurement of retrospective self-report, and we considered this as a bias. This was not a problem for urine toxicology, which is generally considered to be an objective measure. For self-report data, assessors were assumed to be study participants and were aware of the intervention received, hence influenced by social desirability response. For urine toxicology data, we looked for a statement that outcome assessors were blinded to intervention or not. In domain 5, there are instances when in the trial registration, the authors said that they would collect and report urine samples but only reported self-report outcomes in the published paper. Often the rationale for this particular selection were not described. Sometimes we found authors reported more outcomes than they had planned to do. So the suggestions were to adapt RoB2 to allow for multiple assessments of a single study, provide guidance on reporting multiple assessments for studies. Third challenge was interpreting statistics and trial methodology. This was a challenge because RoB2 demands sophisticated understanding of statistics and methodological knowledge. Limited information provided about the methodology in the trial makes assessments difficult. We had to refer multiple records of the trial such as protocol and link records to get more information, and often we could not find protocols or linked records about a trial. We had to make assumptions, and several gray areas were there in these trials. So one of the area was regarding awareness of intervention by the participants and people delivering the intervention. So we had to make an assumption about this area, that if the people who are delivering the interventions, if their input is required, that means the trial is an open trial, and if it is fully automated, then we consider that there is no bias. Most trials did not have published protocol to assess deviations from the intended intervention, and several statistical models dealing with missing data and incomplete reporting was another challenge. So we spent considerable amount of time interpreting statistics and trial methodology. We consulted expert, revisited guidance documents and videos, and we had extensive discussion within this team. So the suggestions we would like to offer is more examples and scenarios should be included in guidance about complex social interventions, particularly around how to assess RoB2 when information is missing or when advanced statistical methods are used. The last challenge, difficulty navigating the guidance. We found the crib sheet lacked sufficient detail, hence very often we had to refer to full guidance document as well as YouTube training videos and webinars. So we did extensive piloting and consensus building exercise. We maintain meticulous record of decisions, and also we developed a review specific guidance document. So I would like to provide some examples on developing review specific guidance document based on our review context. So some of the items is mentioned in this table and how we translated for review specific guidance document. So in domain 3, the item 3.2, is there evidence that the result was not biased by missing outcome data? In this review, we felt that imputing missing outcome data as positive drug use is an acceptable way to correct bias. So we considered simple imputation as an acceptable way to correct the bias. We assume that missing data will be non-random. That is, it is highly likely be due to participant having used drugs or alcohol. Another example from another item 4.2, could measurement or ascertainment of the outcome have differed between intervention group when intervention involves monitoring drug use and control arm doesn't monitor use? This could affect measurement if retrospective self-report data is collected. Another item, is it likely that assessment of the outcome was influenced by knowledge of intervention received? We translated this as for self-report, it would require verification from biochemical marker such as urine toxicology to make an assessment. So unless self-reports are verified by urine toxicology, then we answer no information to assess how likely the assessment of the outcome was influenced by the knowledge of intervention received. So the suggestion was to consolidate guidance into a single multi-layered resource, making it easier to delve deeper into explanations and examples when needed. In conclusion, application of RoB2 is challenging and time-consuming even for an experienced review team. Broad and complex reviews may be particularly susceptible to these challenges. We propose user-friendly ways to revise the RoB2 tool and guidance documents. So these are the references, and we would like to acknowledge the review which was commissioned by the National Institute of Health Research Policy Research Program for the Department of Health and Social Care. Thank you for watching this video.